we will have a webinar, but I will share just the information for today. So I would like, so today is a part of the Kurita Overseas Research Grant. So, but before the information session about the Kurita grant, would like to have the guest lecture by Dr. Kitajima. So, so the lecture will be about 1 hour, and it might be a little bit more. It will depend on the Q&A. And so this webinar, then after the lecture and then Q&A, we have the breakout rooms for participants from Indonesia and participants from Vietnam. So so the information on the Kurita Overseas Grant will be in the 2 languages. So that by Indonesians, by myself, and Professor Viet An will give information on the grant in Vietnamese. So that will be a breakout rooms. So room 1 is for Indonesian, and then for Vietnamese room 2. So that this webinar also part of the that one is the project with the Asia Joint Research Program, the Asia GRP with the BRIN, with the topic of our research within ITB research and also with the Hokkaido and Yamanashi and also with the Vietnamese institutions, that one regarding the wastewater informed early warning system to minimize impact of COVID-19 and also this, it might be other diseases. So I would like just briefly introduce Professor Masaaki Kitajima. He is got the doctoral engineering degree in the field of the urban and environmental engineering in the University of Tokyo, and also he has been in a number of the overseas engagement, either with the University of the Arizona. He did the postdoc, and also in the Singapore MIT Alliance for Research and Technology Center in Singapore. Now he is now an associate professor at Hokkaido University. He wrote a lot of publications with the H index. Currently, last week was about 33, and his research area is cover a broad aspect of the health-related water microbiology from an environmental engineering perspective. So that one is a 2-day lecture by the Dr. Masaaki Kitajima, and I think in the next week, at the end of next week, there will be another lecture. I think so. Dr. Kitajima, now this person.
Yes, so, I know him very well.
It's the same from the same graduated from the same university. Yes, yes. Dr. Rio Honda from Kanazawa. But the topic is still tentative about the risk of the COVID-19 virus in the natural water body receiving wastewater. But that one is we would like to discuss the definitive topic. So that one next year in Thursday, May 6th in the morning time. So without the further ado, I would like to welcome Professor Dr. Kitajima to present your lecture. I think Dr. Kitajima has been assigned as a co-host.
Yes, already.
Yes.
Okay, thank you.
Okay, thank you very much for your kind introduction, Professor Setiadi. So let me start my presentation. Sorry. Okay, so I'm very honored to be here, and so I'm Masaaki Kitajima from Hokkaido University. I'm a associate professor at the Department of Environmental Engineering. And today I'm going to be talking about wastewater-based epidemiology to monitor COVID-19. So I guess you all know about COVID-19, coronavirus disease 2013, right? So let me begin with an explanation of COVID-19 and the causative viral agent. So the causative viral agent of COVID-19 is called novel coronavirus, right? As many of you know. And the official name of this virus is severe acute respiratory syndrome coronavirus 2. The acronym is SARS-CoV-2. So if I mention SARS-CoV-2, please understand that this is the name of the novel coronavirus, official name of coronavirus. So this is the name of the virus of the COVID-19. And the COVID-19 is not the virus, but the name of the disease caused by SARS coronavirus 2. So COVID-19 means coronavirus disease 2019. So for example, what we detect from wastewater is SARS-CoV-2 rather than COVID-19. Okay, so this is a difference between SARS-CoV-2 and COVID-19. And we call the SARS-CoV-2 as novel coronavirus currently, but there was some novel coronavirus in the past. There are two examples. The first example is SARS coronavirus. This is the virus that caused severe acute respiratory syndrome in 2003, which caused outbreaks in China and Hong Kong. And this may be called SARS coronavirus 1 to distinguish between SARS coronavirus and the current novel coronavirus, SARS coronavirus 2. And so the disease caused by this virus is SARS. And the second example is MERS coronavirus. So MERS stands for Middle East Respiratory Syndrome. And this caused outbreaks in 2013 in Middle Eastern countries. So the disease caused by this virus is MERS. And so these are novel coronaviruses, and the current coronavirus is SARS coronavirus 2. So the reason why we call it SARS coronavirus 2 is that this virus is closely related to SARS coronavirus, which emerged in 2003. So this is a phylogenetic tree, and as you can see here, SARS coronavirus 2, which is current novel coronavirus, is closely related to SARS coronavirus, and MERS coronavirus is here. And there are 4 human common cold coronaviruses, which are indicated with orange color here. And the nucleotide identities between SARS coronavirus 2 and other coronaviruses are shown here. And the SARS coronavirus 2 is closely related to bat coronavirus with 96% nucleotide identities. Pangolin coronavirus, 91%. SARS coronavirus 1, original SARS coronavirus with 80% identity. With MERS, only 55%. And common cold coronavirus, which are indicated here, only 50%. So this is the phylogeny of SARS coronavirus 2. So please remember, SARS coronavirus 2 is genetically closely related to SARS coronavirus, which emerged in 2003. Now, let me talk about the wastewater-based epidemiology. So this is an innovative disease surveillance tool for SARS coronavirus or COVID-19 and other viral infectious diseases. So as you may know, current epidemiological approach is based on clinical diagnosis, basically with PCR tests targeting individuals, right? So, but PCR test has a few limitations or issues. The first issue is limitations of PCR capacity due to the cost, experimental capacity and technician and so on. The second issue is only symptomatic infections can be identified by clinical diagnosis because this test usually targets only symptomatic individuals. And the third one is a stigma issue for positive individuals. There will be some discriminations for positive individuals. This is like a major social issue for COVID-19. On the other hand, wastewater-based epidemiology can overcome multiple issues of clinical diagnosis because wastewater contains viruses excreted from asymptomatic individuals in addition to symptomatic individuals. And wastewater sample contains viruses excreted from multiple people, many people. There was some comment here. Chat.
So I think we will if there's any question, I will read for you.
Okay. I will go ahead. And the
yes,
third,
thank you, please.
Yeah, okay. So the third issue, the stigma for positive individuals, can be overcome by wastewater-based epidemiology because wastewater-based epidemiology can understand epidemiology without personal information. So, and our goal is to establish early warning and mass diagnosis infrastructure based on wastewater-based epidemiology. And this can be applicable to municipal wastewater. This is a wastewater treatment plant to understand the disease prevalence in the city. And second, facility wastewater is second example. And by applying facility wastewater, by applying this technology to facility wastewater, we can understand facility level disease prevalence. And this can be a very efficient mass diagnosis system for facility. And then we can also apply this technology to international flight toilet wastewater to understand infection among immigrants. How many people are incoming to our country with COVID-19. In future, we can apply metagenomics approach to identify the virus from wastewater to help early findings of viruses, early identification of the pandemic virus from wastewater samples. Actually, this wastewater diagnosis is not the replacement of the ongoing clinical diagnosis, but this will provide information to complement the clinical diagnosis, which will enhance the PCR testing efficiency. And wastewater data will complement the epidemiological data obtained from clinical diagnosis. So this is the concept of this study. And so actually, we are the first ones who proposed wastewater-based epidemiology as a peer-reviewed publication last year. So this, we wrote a peer-reviewed review paper on SARS coronavirus 2 in wastewater last year. This is the paper. And we started drafting this paper last year, March, so more than 1 year ago. At that time, there was no report on detection of this virus in wastewater. And actually, this paper was accepted just 1 year ago, on April 26th. Today is 27th, so more than 1 year ago. And then at that time, there were only 5 papers, including preprints, reporting the detection of SARS coronavirus 2 in wastewater from all over the world. So then at this time, we got our paper got accepted on our paper on review was accepted. In this review paper, we propose the importance and potential of wastewater-based epidemiology, WBE, to better understand the disease prevalence. For this paper, we published press release from Hokkaido University. So this was what happened last year. And this is the content of the paper. Our paper is a comprehensive review covering the aspects from occurrence in wastewater to risk assessment. And first, we reviewed gastrointestinal symptoms of COVID-19 and shedding of this virus in excreta in feces and urine. And we reviewed evidence for the presence of SARS coronavirus 2 and related coronavirus in wastewater. And third, we reviewed what we proposed that we can understand COVID-19 epidemiology through wastewater surveillance. Then we reviewed methods for SARS coronavirus 2 detection in wastewater, which is very useful for the current research because we have to use some method to detect this virus from wastewater. We also reviewed survival in inactivation of coronavirus and other enveloped surrogate viruses in water and wastewater matrices. And we also reviewed risk analysis or risk assessment on related viruses like respiratory viruses in wastewater and the occupational risks. And maybe some of you may know QMRA, quantitative microbial risk assessment for relevant viruses. And we also reviewed dose response of SARS coronavirus 2, although the data for dose response of SARS coronavirus 2 is limited and relevant respiratory viruses. Now, let me explain the details of the advantages of WBE for COVID-19. So the presence of the viral RNA in wastewater means many things. First, we can trace the circulation of viruses in the community, which provides opportunities to estimate their prevalence, genetic diversity, and geographic distribution. And this makes it possible to monitor the epidemiology of virus infections, even if they are not evident by clinical surveillance. This means that, including COVID-19, this kind of disease causes asymptomatic as well, infections as well. So in such disease, including COVID-19, wastewater surveillance is very effective because we can understand the actual prevalence of the disease, including asymptomatic individuals' infections. And this provides an unbiased method of evaluating the spread of infection. This is especially important for like Southeast Asian countries, like the developing countries, because in such area, resources for clinical diagnosis like PCR machines or diagnostic systems are limited. And in some regions, reporting systems are unavailable. So wastewater-based surveillance is very effective in such regions because this wastewater surveillance method is very resource efficient and cost effective. And we can identify variations in the circulating strains through phylogenetic analysis. For example, now variants are prevalent all over the world, right? But we can understand the prevalence of the variants through a genetic analysis of SARS coronavirus 2. And we can compare between regions, and we can assess the evolution of the virus genome over time. So this is kind of like impressive, isn't it? Because last year we kind of predicted the prevalence of variants, and we proposed that through a genomic analysis, we can trace the evolution and we can compare the prevalence between regions in this first review paper. And the WBE is also useful as an early warning system to determine if reintroduction of SARS coronavirus 2 had occurred in the community. Or conversely, this is useful as an assessment of whether exposures have decreased sufficiently following public health interventions, such as lockdown, social isolation, and social distancing. And this also provides anonymous epidemiological information on disease prevalence in the community. So WBE can avoid individual stigma, which often results from the clinical diagnosis in COVID-19 outbreak. So combined use with clinical testing, which is basically a PCR test or antigen test, could contribute to significant cost savings. So this is the key message from our paper. Now, let me talk about why SARS-CoV-2 can be present in wastewater. First, this virus can be shed in feces because this virus can replicate in our gut, human gut. There are two scientific evidence of the replication of this virus in human gut. The first example evidence is that the receptor of this virus is expressed on human intestinal cells. The receptor is called angiotensin converting enzyme 2, ACE2. So this is a receptor for SARS coronavirus 2, SARS coronavirus, original SARS coronavirus. That's why this virus can attach to the cells on our epithelial intestinal cells. The second evidence is that this virus can replicate in human gut enterocytes. So this is in vitro study. So in outside our body. So the researchers inoculated this virus into cell culture of human gut enterocytes, and they observed the replication of the virus in vitro. And they they think that this virus can replicate in vivo in our body as well. So actually, this virus can be shed in feces. There are many reports on SARS coronavirus 2 RNA detection in feces. One example is here. And the viral load or viral concentration can be up to 10 to the 8th copies per liter, per gram feces. So this is quite high concentration. And the shedding of the virus into feces can continue for approximately 4 weeks. And approximately 2/3 of the patients shed the virus, 2/3, so like 66%. On the other hand, approximately only 6% of the patients shed the virus in urine. This means that probably the majority of the virus in sewage or wastewater is originated from feces rather than urine. Okay. So, and another thing is, I keep saying through wastewater surveillance, we can understand the infection status of asymptomatic individuals as well. Actually, SARS coronavirus 2 RNA was detected in feces of asymptomatic individuals. There is scientific evidence. And the ratio of asymptomatic infection is up to 30%. So this is not negligible. So a quite high percentage of the infected individuals are asymptomatic. They don't show symptoms. And another concern is whether an infectious virus can be present in wastewater. Actually, infectious SARS coronavirus 2 was detected in feces. So it is suggested that fecal oral transmission is possible. But we don't have any scientific evidence on the presence of infectious SARS coronavirus 2 in wastewater at the moment. So this is the methodology of COVID-19 or wastewater-based epidemiology. If the COVID-19 is prevalent in the city, the wastewater contains viruses, SARS-CoV-2, and we can detect SARS-CoV-2 in wastewater after we concentrate the virus in wastewater. We use qPCR technique to detect the virus in wastewater, but the concentration of the virus in wastewater is usually very low. So we have to concentrate the virus so we can detect the virus genome by qPCR. So we can concentrate the virus using various methods, and we extract RNA. We extract RNA, and then we detect and quantify the RNA of SARS-CoV-2. So RNA is the genetic material of SARS coronavirus 2. So in order to detect virus in wastewater, we have to concentrate the virus, as I mentioned earlier. But there was no standardized protocol for SARS coronavirus 2 detection from wastewater. So we needed to explore the method to detect SARS-CoV-2 from wastewater efficiently. And so the reason why we needed to explore the new method was that was the difference due to the difference between SARS-CoV-2 and enteric viruses, which were major targets for environmental virologists before pandemic era, COVID-19 pandemic era. And specifically, enteric virus has a capsid protein. This is the outmost layer of the virus particles. On the other hand, SARS coronavirus 2 has lipid envelope. That's why, so these two types of viruses have different in size, hydrophobicity. So as you can imagine, envelope is more hydrophobic, and stability under extreme pH may be different as well. Probably the SARS-CoV-2 is more fragile as compared to enteric virus, like norovirus. So because of these reasons, there was difficulty to apply the existing virus concentration methods for SARS-CoV-2. That's why we evaluated the coronavirus recovery efficiency from wastewater. For this experiment, we inoculated murine hepatitis virus, MHV, which is not infectious to humans, but infectious to mice. And then, we seeded this virus into wastewater aliquots, and we recovered the inoculated virus using 7 different methods based on electronegative membrane, ultrafiltration, PEG precipitation, polyethylene glycol precipitation, and centrifugation. And then we enumerated MHV by RT-qPCR, and we calculated recovery efficiency. I'm gonna skip the results, and from now, let me show you some examples of the actual detection results of SARS-CoV-2 RNA from wastewater samples. This is the first study that reported the detection of SARS coronavirus 2 RNA from wastewater in the world as the peer-reviewed paper. This study was reported from Australia, and I also contributed to this study. In this study, we collected wastewater samples from 3 wastewater treatment plants in Brisbane, Australia, and we concentrated the virus using HA, electronegative membrane, and we detected the virus using 2 different qPCR assays. And this is the results. And this is kind of a busy figure, but in the summary of the result is that we detected SARS-CoV-2 RNA from wastewater samples during outbreak period in this region. And the viral RNA concentration was up to 120 copies per liter. and we confirmed qPCR product sequence. So this was indeed, the signal was indeed originated from or derived from SARS-CoV-2 genome. And based on this concentration, we estimated the infection prevalence. And the median value was 0.096%. So this means roughly out of 1,000 inhabitants or population, one person got infected. So 1 out of 1,000 people got infected with this virus. This was our calculation. And so we also reported the recovery efficiency of coronavirus in wastewater. Sorry. So this was associated with this slide. Sorry, but yeah, we also reported the recovery efficiency, and we published press release from our university in last August. We also detected this virus RNA from wastewater samples in Japan as well. So this is the paper, and I also contributed to this study. In this study, we collected wastewater samples from influent, secondary treated sewage, and river water samples from wastewater treatment plant and river. And we concentrated the virus, and we detected SARS-CoV-2 using 6 different assays. So here's the summaries of the results. And we detected the virus from the wastewater samples collected from April 14 last year. And in this period, the number of infected people increased in this region. So this also indicates that wastewater-based epidemiology can grasp the prevalence of the disease in a certain region. And this may be applicable to Japan as well. Okay, and then we also detected the virus in wastewater in North America for the first time. And this is, in this study, we collected wastewater samples from 2 wastewater treatment plants in Louisiana, which is located in southern United States. And for this result, we also published press release from our university. So this is a brief summary of the results. Looking at this figure, the filled circles indicates the detection of the virus, positive samples from influent wastewater samples. This also indicates that virus can be detected from wastewater when the number of infected people increases in each region, in the region. Actually, Louisiana was one of the most significantly affected states in the United States in the first epidemic period. And this is the first detection of SARS coronavirus 2 RNA in wastewater in North America, including United States. So actually, I showed you the first detection in Japan, right? So this was earlier than this report, which means that our report from Japan preceded the first report from the United States. And I was associated with both publications. And SARS coronavirus 2 RNA was detected in wastewater immediately after COVID-19 outbreak in this region. And so until now, I've been talking about application of wastewater-based epidemiology to municipal wastewater samples, but this approach can be applicable to aircraft and cruise ship, so like mass transportation systems. So this wastewater surveillance can be a potential tool to understand imported COVID-19 infections for when we apply this approach to international flight and international cruise ship. So basically the method to detect virus from those wastewater samples is the same as the that is used for municipal wastewater samples. But the main message here is that this can be one of the tools to understand how many people are incoming to a specific country with COVID-19 infections. Then we also evaluated the persistence of SARS coronavirus 2 in wastewater using culture method, cell culture method. We conducted this experiment using infectious SARS coronavirus 2 in BSL-4 facility in the United States, and we have already published this paper. And in this paper, we inoculated infectious coronavirus 2 in tap water and wastewater, and we incubated these inoculated samples at different temperatures. And we had two different titers, inoculation titers, and we titered the virus concentration using cell culture, and we estimated the decay rate constant. On the other hand, we also quantified the viral RNA using RT-qPCR, and we also estimated the first order RNA decay constant. So these are the results, and I'm not going to have time to walk you through the details of the results, but this research determined the activation rates of SARS coronavirus 2 in wastewater under different temperatures. So temperature is the major determinant of the survival of the virus in wastewater samples. And this study revealed persistence of SARS coronavirus 2 in wastewater, and this is important data for risk assessment. And we also evaluate the decay of RNA signal in wastewater. We call it RNA signal because viral RNA is indicative of viral disease prevalence in a specific region. That's why we call it RNA signal. And we are detecting RNA rather than infectious RNA, infectious virus for wastewater-based epidemiology studies, WBE studies. In this study, we determined decay rate constants of SARS coronavirus 2 and murine hepatitis virus RNA in water and wastewater. And this is the results in these panels. We showed decay rate of viral RNA under different temperature for SARS coronavirus 2 and MHV. For untreated wastewater, autoclave wastewater, which doesn't have biological activity, and tap water samples. And the summary of the result is here. These two figures summarizes the viral RNA decay constants and environmental factors that may impact the survival, or not survival, but RNA decay rates. So looking at this figure, so the major determinants of the RNA decay rate is temperature rather than the biological activity or sample matrix type, tap water or wastewater. Temperature is the major determinant. That's what we obtained from this study. So from now on, let me talk about our collaboration with Shionogi, a company in Japan. Shionogi is the major pharmaceutical company in Japan, and we are now actively collaborating with this company. And we are conducting technological development and societal implementation of wastewater-based epidemiology of COVID-19 in Japan. And this is one of the publications that we already released through foreign media. Please look at this site if you are interested. And there are a few upcoming articles from this news media as well. And through this collaboration, we developed a highly sensitive method for virus SARS-CoV-2 detection in wastewater samples. Let me explain some details. So in this study, we developed a new method, but in conventional method, we used to use pre-treatment. So I guess many of you are environmental engineer or environmental scientist, and some of you may be doing some chemical analysis or microbial analysis of water quality. But in water quality analysis, we usually remove the particles or suspended solids in wastewater because they inhibit the analysis. So in conventional method, we use pre-treatment. That means we used to remove the solids from wastewater before we concentrate viruses. And we concentrate the virus from water fraction, and we detect it with qPCR. But we realized that this method is not very effective for SARS-CoV-2, although this is effective for other viruses like human enteric viruses, including norovirus and other viruses. But for novel coronavirus or SARS-CoV-2, this method is not efficient. That's why we developed a new method which does not include pre-treatment. So we concentrated the virus with solids, and we invented a method to extract RNA efficiently from the solids. And we do pre-amplification of the genome before we quantify the genome using qPCR. So basically, we quantify the amplified DNA fragments for sensitive detection. So this allows sensitive detection, but we can still quantify the number of the viral RNA in the original wastewater samples. And then we also developed a qPCR assay for variants identification. So we showed that we can detect SARS-CoV-2 variants from wastewater using next generation sequencing or NGS. So this is the map of the SARS-CoV-2 genome. The SARS-CoV-2 has a length of around 30,000 base pairs, and this has like many open reading frames. The important region for variant analysis is S or spike region, which encodes spike protein of the viral particle. So spike protein is associated with attachment of the virus particle to receptor ACE2. So if the amino acid mutates to another amino acid on the spike protein, this may change the phenotype of the virus, or in other words, this may increase the infectivity or transmissibility, or this may reduce the vaccine efficiency. So there are some like important amino acid mutations as listed here, E454K, which reduces vaccine efficiency or escape from immunity. N501Y is a unique amino acid mutation of the variants, which increases infectivity. And D614G is associated with increased replication efficiency and transmissibility. So this is the very important region. That's why we wanted to analyze this region, but the existing existing nested PCR methods did not target this region. So we couldn't analyze the genome sequence in this region. So we developed PCR for variant identification. So this is an original assay, which allows identification of amino acid mutations among variants. And this is suitable for Illumina pairing sequencing. And this is the result of the next generation sequencing library preparation. So this is a result of the library prep. We obtained PCR product and we quantified the PCR product and we did library preparation and we sequenced the genome using MySeq from Illumina. And we did in situ analysis to identify the mutations. And this is the result of bioanalyzer. And we, as you can see here, library preparation was successful for NGS analysis. And this is the result. We obtained high quality NGS reads of approximately this number, like up to closely 1 million reads per sample. So this is a ultra deep sequencing. And we detected N501Y, D614G, and these are commonly observed among variants. And so this is a result of the quarantine hotel and wastewater influent in Japan. For quarantine hotel, we detected variants in December, but this variant was not detected from October and November, which is reasonable because in Japan, we identify these variants in December, not in October and November. And then we applied this technique to domestic or municipal wastewater. And this is a result. And in municipal wastewater, we detected this variant from the samples collected in December 4th, which is kind of surprising because Japanese government first reported the detection or identification of these variants on December 25th. So 3 weeks before the first report from the government, we detected this variant from wastewater samples. And then the proportions of the reads of the N501Y variants increased from December to January. So this is a significant findings. And yes, and 501Y mutation detected in municipal wastewater earlier than the first confirmed case in Japan. So we showed the evidence of the application of this virus detection in laboratory. So now the time to implement this technology to the society. And we achieved full automation of virus detection. And this is a press release on the establishment of an automated system for the analysis of SARS-CoV-2 in wastewater. And this system involves robotics and high performance or high throughput next generation sequencing system. We actually use a lab droid, Mahoro, which is a versatile humanoid robot made in Japan. So RBI, Robotic Biology Institute, has this robot. And this robot does everything from virus concentration to PCR plate preparation. And also, this robot does library preparation for next generation sequencing. And we also collaborate with ILAC, which has Illumina NovaSeq platform, which allows high throughput sequencing analysis. For this societal implementation, this was picked up by or featured by international media, Innovation News Network, last month. And we also started monitoring in Osaka Prefecture to monitor COVID-19 through wastewater surveillance. And so this monitoring activity was, for this study, we started this month. And so this is the last topic, actually. So we are going to hold the Summer Olympic Games in Tokyo, as you may know, right? But probably the COVID-19 will be still there, and we have to have Olympic Games with COVID-19. So risk management of COVID-19 during the Tokyo Olympic Games is very important, and this is primary responsibility for relevant Japanese researchers, we think. And I think I'm one of the responsible researcher in Japan. And wastewater testing can be used as one of the tools to manage risks. When we apply this testing tool to the Olympic athlete village in Tokyo to manage the risks. So this is summary of my talk today. So WBE is very useful for viral disease, viral infectious diseases. And WBE was previously applied to norovirus, poliovirus, and so on, basically enteric viruses. And applicability of WBE to COVID-19 was proposed by our paper for the first time. So this paper was the first review paper proposed the concept of WBE. Currently, there are a number of reports on the detection of SARS coronavirus 2, including variants from wastewater. So now the concept that we proposed in our first review paper has been proven. So now we have to implement this. And I'd like to highlight that the WBE can add the value of wastewater infrastructure. Specifically in the textbook, I guess many of you are environmental engineer. So if you open the textbook of environmental engineering, it is written that the wastewater infrastructure was constructed primarily to treat and remove wastewater and stormwater, right? But in this time, the new role, which is monitoring of disease prevalence, is being added to the value or role of wastewater infrastructure. So this is a very important time that changes the image and the value of the wastewater infrastructure. Now, what we need is to implement this infrastructure or technique to the society. So societal implementation is urgently needed. With that, I would like to thank you for your kind attention, and I'll be very happy to take any questions.
Okay, thank you, Dr. Kitajima. So that, thank you very much. So if you have, I think so, if you have any question, you may write in the chat box, or you can open your mic and then raise your question directly. So we still have some time. So please, if you have any question and comment or this is, yeah, thank you. So that please, if you have any question, or so I think so the question from myself, Dr. Kitajima. So that if you mentioned that the variant can be detected earlier before the cases has been identified in Japan. Mm-hmm. So that one was, you realize after the cases occur, right? So in the beginning, you already realized that something, because in the review you have mentioned that the variant can be detected.
That's right. That's right.
But that in the cases, in the real cases, where when your analysis, so that one is you realize that it is happened what you have predicted. True.
Yeah. So yeah. Let me clarify if I understand your question correctly. So we actually detected variants from archived samples. So we So
I see.
started viral analysis in this year, and so we analyzed stored sample or frozen samples, and we
I
went
see.
back to the older dates, and then we successfully identified the variants from the December 4th sample,
I
so which
see.
which was preceded to the first detection or first report from the Japanese government on the variants. Yeah. So I hope this answers your question.
Yes, yes. So that one is
Okay.
uh, you you store the sample and then
That's right. That's
uh,
right. We froze the sample and we
okay,
analyzed
okay,
frozen samples.
okay, okay,
Yeah, yeah.
okay. Please, if you have any other question, you may raise directly to Dr. Kitajima. Please.
There is one question, Professor Chandra.
Oh, one question. Okay, okay. Guntur, could you help me? Because I have.
Yes.
Please.
Wait a bit.
Because I have to read.
So I can read the question.
Maybe.
Yeah, yeah. Please kindly share thoughts on how far we can develop and apply that automatic monitoring station for from today.
That one is from Vietnam.
Okay, okay. Oh, yeah, yeah, yeah. Okay, so automatic monitoring station. Actually, we have achieved.
Yes.
So we are currently very close to the position where we can develop automatic monitoring station, because now Japanese lab droid, like robot, versatile humanoid robot is available for wastewater sample processing and detection. So if we can install this to like many stations, then we can use this technique. So technically, this is feasible. But the issue is the cost, actually. This costs $1 billion, $1 million per robot.
Oh.
So this is quite expensive, quite expensive. But we have already, technically, we have already established this monitoring station. So if we can reduce the cost, probably this is feasible. Yeah, probably this automatic monitoring station can be realized. And how far? In Japan, we have already such station now, but this is only available only one station in Japan. So my goal is to expand this station or make duplicates or triplicates, copies of this station in all over Japan and all over the world. This is my goal. And another thing, we were able to establish automatic monitoring station because the robot is versatile, right? So this is not the robot specific, so special robot for a certain process, but this robot can do anything that humans can do. Okay, so this robot has 2 arms, and this robot has the freedom of the freedom degree of seven freedom of degrees. So this can do anything that we can do, like pipetting or like open the tubes or open micro tubes. This robot can do anything. So that's why we have been successful in developing automated system. Otherwise, this may take, the robotic expert says, this may take like 5 years if the versatile robot is not applicable or is not available. So yeah, I think how far in Japan, we have already established, and we will provide the service of the wastewater analysis next month, actually, which is impressive, isn't it? Using the robot. So this robot can work 24 hours, right? Of course, and 30, 365 days, all day, around the year. So this may speed up the wastewater analysis, and this will provide very high throughput of the wastewater analysis. Yeah, I hope this answers your question.
Okay. So that one is this.
Yeah. Okay. So let me answer Miss Gunter's question. So yes, actually, there's no experimental evidence that detected infectious SARS-CoV-2 novel coronavirus from wastewater, but there's no experimental evidence. But there is epidemiological evidence, which means that in China. So this is one example. In China, there was an outbreak in apartment complex, right? And in that apartment complex, there was a failure in drainage system, wastewater drainage system. Okay, and then the wastewater spilled from the failure, and the spill out, and probably the aerosol was generated, and this the people got exposed to the drain wastewater, and then the people who got infected was associated with exposure to wastewater, which means that probably wastewater was the source of the virus transmission from epidemiological evidence. So there is one. So this is one example, but there is one. There are some epidemiological evidence of transmission of the virus through wastewater, but there is no experimental evidence that successfully cultured infectious SARS-CoV-2 from wastewater. But so what I would like to say is, if you get exposed to fresh wastewater with the virus, you may get infected from the wastewater. But there is no evident risk of the wastewater exposure at the wastewater treatment plants. So there is no example of the exposure and establishment of infection at wastewater treatment plant. So that means probably the risk of the exposure to wastewater at the wastewater treatment plant is very low. I think this is negligible. Probably because, like you said here, the lifetime of the virus in wastewater is very short. So the virus doesn't survive in wastewater very long. So this was indicated by our study. So in my presentation slide, let me show the slide. I hope this answers your question here. So the latter part of your question. So how long the virus survives in wastewater, right? So in this paper, you can find the information of the survival of the virus in wastewater. Yeah. So this is the TCID, which is associated with the infectious virus titer result, the upper panel. So the virus titer decreased very rapidly at room temperature. And this is the Yeah. This is the RNA. This is RNA. This is the infectious titer, but the infectious titer decrease very rapidly. So the virus doesn't survive in wastewater very much, and this is temperature dependent. The higher the temperature, the higher the inactivation speed.
Hmm.
So yeah. So this is, I guess, one example, and you can find this paper, and this is downloadable. So you can download this paper PDF from the Environmental Science and Technology Letters website.
Okay.
Yeah, if you don't have PDF, I can send you the copy of the PDF. I hope this answers your question. Yeah.
Okay. Is there any other question from the audience? Oh, there's a field Datun, please.
Yeah, good afternoon, Associate Professor Masaaki Kitajima. Thank you very much for the presentation, very useful information. I'm Frida Tunisa, based in Surabaya with ITS. Just have a quick question related to the technical part of the sampling of the wastewater. So we understand that this coronavirus, we could eventually consider this virus as a pathogen. So that means, do we treat the people who take the wastewater sample like we normally take the sampling, like just a normal protection, or is there any specific consideration for that? That the first one. And then the second related to, because in your presentation you mentioned regarding the needs of the concentration to be concentrated the sample. Is there any specific time range of the day, like the best time that we could take the sample from the, for example, from the wastewater treatment plant? For example, in the morning where most of the people use the toilet. That the second. And then if we take the sample in the morning, do we still need to concentrate it at the sample again, even though we understand that we probably will find more sludge in the morning? That's my question.
Okay. Thank you for your question. Yeah, very good questions. So the first question, so the PPE, right? Personal protective equipment. How we have to cautious about the infection when we collect wastewater samples. Basically, the normal PPE is enough for wastewater sampling, because in municipal wastewater, SARS-CoV-2 concentration is relatively low, and normal PPE is enough. But I would suggest you wear gloves, goggles, and the gowns at least when you collect samples. But other than that, that's it. Yeah. So the second question is sampling, right?
Yeah, the time range regarding the concentration.
That's right. That's right. Yeah. Okay. So as you may imagine, the virus concentration fluctuates over time within a day, right? At the wastewater treatment plant. If you have automated sampler, you can collect composite sample, or you can do 24-hour continuous sampling to identify the best time of the day to collect for efficient wastewater testing. So I remember your question. So whether we can detect virus without concentration, right? So in the United States, for example, they can detect virus. They could detect virus without concentration. When that means they directly extracted viral RNA from the wastewater samples without concentration, and they could detect the viral RNA using qPCR when the COVID-19 was very prevalent in the United States. But the number of the cases is rapidly decreasing in the United States. Now the detection rate is decreasing. So now the concentration of the virus level, virus concentration is below the detection threshold where they can detect virus without concentration, but certain period of time they could detect virus detection. So depending on the prevalence, you could detect the SARS-CoV-2 RNA without concentration. But in many cases, including Japan, you will need to concentrate the virus to detect viral RNA from wastewater. And for the time variation, we propose that you collect composite sample, 24-hour composite sample, or if the automated sampler is available. But if auto sampler is not available and you need to collect grab sample, probably the first the morning is the best time. I agree with you. And actually, we are collecting grab samples as well, and we are collecting around 10 a.m. considering the time that when people go to the toilet and resident time in sewer. And we also monitored the virus level in wastewater, and we observed that in the morning, virus concentration is relatively high, but in major cities, the catchment area is very high, very wide, and there are certain several hours residence time. So the virus concentration is kind of normalized over time. So you can collect in the morning, but it doesn't. Well, it may affect, but it depends on the size of the catchment and depends on the treatment plant. So
can I add something?
Hmm?
Yeah, is it also related to the temperature, for example, in Japan? Because I think, if I'm not mistaken, that you need to treat the sample below 4 degrees Celsius for the handling before you analyze the sample. Or we could what is your suggestion how we transport the sample? Do we need to put in the S box and something like that?
Yeah. So when we transport the samples, we transport on ice or keep refrigerated. If the virus, the sample arrives within a day or two, but for longer transportation time, we froze the sample, and we freeze the sample and then transport and then defrost upon arrival.
Okay, yeah. Thank you very much, Professor Masaaki.
Yeah, no problem.
Okay, I think so. The last one, because of the time, Guntur, probably this one in the chat box is from. Okay, so that one, the last one from, I think.
Okay. Lagging time of addiction. This is good point. Also, a very good point. Okay, so let me explain how the early detection is possible through wastewater surveillance. So when one person get infected with virus, there is certain incubation period, right? Incubation period is called, on average, 2 weeks, you may have heard. Within 2 weeks, there is a certain point where the virus shedding starts in feces. And then after a few days to several days, the person start exhibiting symptoms. So by PCR tests, in other words, a clinical diagnosis, we can identify the infection after the infected individual exhibit symptoms, right? Like fever or cough and so on. And then they get reported. But through wastewater surveillance, we can identify the virus from wastewater before the person exhibit symptoms. So before they exhibit symptoms, they start shedding the virus several days earlier than when they exhibit symptoms. That's why we can if we routinely and continuously monitor the wastewater for SARS-CoV-2, we have the possibility that we can identify the virus before the increase in the reported cases. I hope this answers your question.
Okay. Okay, I think so.
Okay, great.
Okay, thank you. So I think so. Would like to end our session. We'd like to thank Dr. Kitajima for you very interesting presentation. I think hopefully this information will be useful, especially some of the audience here is the young researchers. So hopefully they'll encourage the young researcher to do the research either on the epidemiology or it might be in the wastewater treatment, wastewater environment as general. So Guntur, probably you could take a picture of all of us before we are moving.
Good idea.
Yeah. So that I think in the meantime, I would like to give the information that for the young researchers, so there will be 2 rooms. Room 1, it is for the Indonesian, and room 2 is for the Vietnamese. So the Indonesian will goes to the room 1, and then the Vietnamese to the room 2. That one is for the young researcher who are interested into the Kurita grant. So me and will be in the room 1, and Professor Viet An will be in the room 2. Probably